What if I told you that two tiny chemicals in your brain could decide whether you're connecting with the real world or trapped in a distorted one? That the difference between chatting with a friend and answering voices that no one else hears could come down to dopamine and glutamate. Welcome to the Pharmacology Vault, your ultimate spot for turning heavy pharmacology into clear, visual, and easy to remember stories. If you're new here, hit subscribe right now, because today, we're unpacking one of psychiatry's most intriguing mysteries, the dopamine and glutamate hypotheses of schizophrenia. Let's bust a myth straight away. Schizophrenia is not split personality. It's a chronic, severe mental disorder that distorts how a person thinks, feels, and behaves. Here's a quick look at the numbers. Globally, schizophrenia affects about 1% of the population. It usually appears early in life. In men, it tends to start between 15 and 25 years, while in women, it's slightly later, between 25 and 35 years. Research shows that males often experience earlier onset and more severe symptoms. Risk factors? Well, genetics is a big one. If a close relative has it, the chances are higher. But it's not just genes. Obstetric complications during birth, cannabis use during adolescence, and even growing up in large urban cities are all linked to higher risk. When we talk about schizophrenia, we usually classify symptoms into three major domains. Positive symptoms, these are symptoms that add something abnormal to behavior. Think hallucinations, seeing or hearing things that aren't there, and delusions, fixed, false beliefs. Negative symptoms, these are things that are taken away. For example, a flat affect, showing little to no emotional expression, or avolition, a complete lack of motivation. Cognitive symptoms, these affect thinking and memory. People may struggle with working memory, have trouble focusing, or experience executive dysfunction, meaning they find it hard to plan or make decisions. Now, to understand schizophrenia at the brain chemistry level, we need to look at two major players, dopamine and glutamate. First, the dopamine hypothesis. This has been one of the longest-standing theories in psychiatry. It suggests that in schizophrenia, there's too much dopamine activity in the mesolimbic pathway, and this is what drives the positive symptoms like hallucinations and delusions. On the other hand, there's too little dopamine activity in the mesocortical pathway, which is linked to negative symptoms like lack of motivation and flat affect, as well as cognitive deficits like poor working memory. Next, the glutamate hypothesis. This focuses on another key neurotransmitter, glutamate, which is crucial for brain signaling and plasticity. In schizophrenia, there's evidence for NMDA receptor hypofunction, meaning these receptors aren't working as well as they should. This disrupts the cortical-subcortical loops, affecting brain connectivity and potentially contributing to both positive and negative symptoms. So, while the dopamine hypothesis explains a lot about symptoms and medication effects, the glutamate hypothesis gives us a broader perspective, especially for symptoms that dopamine-based drugs don't fully address. Now let's dive deeper into the dopamine hypothesis, which is one of the most well-known neurochemical theories of schizophrenia. First, let's look at the mesolimbic pathway. When dopamine levels here are excessively high, they overstimulate D2 receptors, especially in the nucleus accumbens. This overactivity is strongly linked to positive symptoms, things like hallucinations and delusions. In simple words, the brain starts misinterpreting reality because the reward and salience system is overfiring. Second, the mesocortical pathway tells a different story. Here, dopamine levels are actually reduced, especially in the prefrontal cortex. This understimulation of D1 receptors is linked to negative symptoms like flat emotional expression and social withdrawal, as well as cognitive symptoms such as poor working memory and disorganized thinking. It's almost like two parts of the brain are living in opposite chemical worlds, one with too much dopamine and one with too little. Evidence? Antipsychotic drugs, which block D2 receptors, often lead to a clear reduction in positive symptoms. Stimulants like amphetamines, which increase dopamine release, can actually trigger psychotic-like symptoms in healthy individuals. Brain imaging studies, especially PET scans, show that people with schizophrenia often have increased striatal dopamine synthesis capacity, supporting the hyperdopaminergic aspect of this hypothesis. So, in summary, the dopamine hypothesis gives us a dual perspective. Too much dopamine in the mesolimbic pathway, right pointing arrow, positive symptoms. Too little dopamine in the mesocortical pathway, right pointing arrow, negative and cognitive symptoms. Now let's look at the NMDA hypofunction hypothesis, one of the key neurochemical explanations for schizophrenia. Here's the idea. When NMDA receptor activity drops, the brain's GABAergic interneurons, our main inhibitory control cells, become less effective. That means they can't keep excitatory neurons in check. The result? A glutamate surge. This glutamate overactivity can cause neurotoxicity and also throw dopamine regulation off balance, especially in subcortical areas. That's why we see both positive symptoms, like hallucinations and delusions, and cognitive symptoms, like poor memory and disorganization. Mechanistically, NMDA hypofunction impairs cortical filtering, allows excessive dopamine firing in subcortical regions, and disrupts communication between the prefrontal cortex, thalamus, and hippocampus. What's the evidence? Well, drugs like ketamine and PCP, which block NMDA receptors, can actually mimic the symptoms of schizophrenia in healthy individuals. And in postmortem studies, patients with schizophrenia have fewer NMDA receptor subunits in key brain regions. This makes NMDA hypofunction a very compelling piece of the puzzle. Let's put both major theories of schizophrenia side by side to understand how they differ and where each fits in. Primary neurotransmitter. The dopamine hypothesis focuses on dopamine, suggesting that too much dopamine activity, especially in mesolimbic pathways, drives positive symptoms like hallucinations and delusions. In contrast, the glutamate hypothesis focuses on glutamate, particularly NMDA receptor activity, and how its underfunctioning disrupts brain circuits. Core theory. In dopamine theory, an increase in dopamine activity causes positive symptoms, while a decrease causes negative and cognitive symptoms. But glutamate theory takes a broader view. It proposes that reduced NMDA receptor function triggers a chain reaction, disrupting glutamate, dopamine, and even GABA circuits, leading to widespread dysregulation. Evidence. We know dopamine antagonists, like antipsychotic drugs, reduce positive symptoms, which supports the dopamine theory. On the other hand, NMDA antagonists like ketamine or PCP can actually induce schizophrenia-like symptoms in healthy individuals, supporting the glutamate theory. Scope. The dopamine theory is more narrow, it mostly explains the positive symptoms. The glutamate hypothesis covers more ground, it also explains negative symptoms, cognitive deficits, and the complex interaction between brain circuits. Clinical limitation. Here's the key difference. Dopamine-based treatments often fail to improve negative and cognitive symptoms. Glutamate theory offers a possible explanation for these symptoms and suggests that future treatments may need to go beyond dopamine alone. Let's explore how specific brain regions and neurotransmitter changes contribute to the core symptoms of schizophrenia. Starting with the ventral striatum, particularly the nucleus accumbens, here, there's an increase in dopamine activity, especially at D2 receptors. This overactivity drives the positive symptoms such as hallucinations and delusions, by amplifying reward and salience signals to inappropriate stimuli. Moving to the dorsolateral prefrontal cortex, we see a reduction in dopamine and a decrease in NMDA receptor activity. These deficits impair working memory, attention, and decision-making, explaining the cognitive symptoms that make daily functioning difficult. In the hippocampus, excessive glutamate release leads to disinhibition of downstream pathways. This results in a dopamine surge within the striatum, further fueling psychotic episodes. Finally, the thalamus, a crucial relay station, shows signs of cortical disconnection. This disrupts the brain's ability to filter and process sensory information, leading to sensory misprocessing, where ordinary sounds, sights, or sensations may feel distorted or threatening. Together, these neuroanatomical changes form a complex network of dysfunction, where dopamine, glutamate, and connectivity abnormalities interact to produce the diverse symptoms of schizophrenia. Now, let's talk about how antipsychotics work by targeting dopamine pathways. We have three main drug classes here. First, the first generation, or typical antipsychotics, like haloperidol and chlorpromazine. These work by strongly blocking D2 dopamine receptors. This helps control positive symptoms like hallucinations and delusions. But the downside? Because they block dopamine so strongly in other brain regions, they can cause extrapyramidal side effects, things like muscle stiffness, tremors, and, with long use, tardive dyskinesia. Second, the second generation, or atypical antipsychotics, such as risperidone and olanzapine. These block both D2 receptors and 5-HT2A serotonin receptors. This dual action not only treats positive symptoms but also helps with negative symptoms to some extent, like social withdrawal or reduced emotional expression. They also have lower rates of movement side effects, though they often cause metabolic issues, weight gain, high cholesterol, and diabetes risk. Finally, we have D2 partial agonists, like aripiprazole. These are interesting because they act like a dimmer switch for dopamine, boosting dopamine where it's too low and blocking it where it's too high. This means they can balance dopamine activity rather than just shutting it down completely. But here's the catch: no current drug fully fixes negative or cognitive symptoms, and all have side effects to watch for, whether it's movement problems, hormonal changes, or metabolic risks. So, while dopamine targeting is effective for the core psychotic features, it's not the whole solution. We still need treatments that address the full spectrum of symptoms. In schizophrenia treatment, the glutamatergic system is becoming an exciting new frontier, especially for addressing negative and cognitive symptoms that remain resistant to dopamine-based therapies. Research focuses on modulating the NMDA receptor, AMPA receptor, and metabotropic glutamate receptors, MGLURs, to restore balanced neurotransmission. NMDA enhancers, agents like D-serine and glycine, act as coagonists at the NMDA receptor's glycine site, improving NMDA receptor activity. This can enhance synaptic plasticity and cognition. GlyT1 inhibitors, drugs like bitopertin, inhibit the glycine transporter 1, which prevents glycine reuptake and thereby increases synaptic glycine availability, further boosting NMDA function. AMPA modulators, compounds such as CX516 and CX717 enhance glutamate signaling via AMPA receptors, which may improve neuronal firing and cognitive processing speed. MGLUR agonists, targeting MGLUR 2/3 receptors, reduces excessive presynaptic glutamate release, helping to stabilize excitatory-inhibitory balance in the brain. Anti-inflammatory agents, minocycline, though traditionally an antibiotic, can reduce microglial activation and neuroinflammation, processes that may contribute to symptom persistence. Clinical insight: when used alongside antipsychotics, several of these agents have shown promising effects in improving negative symptoms like social withdrawal and cognitive deficits such as poor memory and attention, areas where traditional dopamine-targeted drugs often fall short. Schizophrenia is increasingly understood as the result of interacting genetic vulnerabilities and environmental stressors rather than a single neurotransmitter imbalance. In this model, genetic predispositions, such as mutations in DISC1, NRG1, and COMT, set the stage for abnormal brain development. When combined with environmental insults like perinatal hypoxia, prenatal or early life infections, or adolescent cannabis use, these vulnerabilities disrupt normal neurodevelopmental processes. A key feature is impaired NMDA receptor function, leading to cortical disinhibition. The brain's top-down control is weakened. This loss of inhibitory regulation allows subcortical dopamine activity, particularly in the mesolimbic pathway, to increase abnormally. This dopaminergic overdrive is linked to positive symptoms such as hallucinations and delusions. During adolescence, excessive or abnormal synaptic pruning further exacerbates neural network dysfunction, locking in a trajectory toward the full-blown schizophrenia phenotype. Importantly, the model integrates the dopamine and glutamate hypotheses, showing they are partners, not rivals, in the disease process. Dysfunctions in glutamate signaling can drive dopaminergic abnormalities, creating a self-reinforcing neurochemical loop that underpins both positive and negative symptoms, as well as cognitive deficits. Let's wrap up by pulling all the threads together. Positive symptoms. In schizophrenia, positive symptoms, like hallucinations and delusions, are strongly linked to excess dopamine activity in the mesolimbic pathway. This overdrive of dopamine disrupts reality perception, creating vivid but false experiences. Negative and cognitive symptoms. On the other hand, symptoms like social withdrawal, lack of motivation, and impaired working memory trace back to reduced dopamine and NMDA receptor function in the prefrontal cortex, along with hippocampal dysfunction. These changes dampen higher-order thinking and emotional regulation. Dopamine hypothesis. This classic model proposes that overactivity of D2 receptors in certain brain circuits drives psychosis. It's the foundation for why most current antipsychotics work by blocking D2 receptors. Glutamate hypothesis. Here, the focus shifts to NMDA receptor hypofunction. When these receptors underperform, cortical circuits lose inhibitory control, triggering a surge in subcortical dopamine. This bridges the gap between the glutamate and dopamine theories. Treatment approach. Today's treatments still rely heavily on D2 blockade to control positive symptoms, but the future lies in glutamatergic modulation, finding ways to restore NMDA receptor activity for a more complete symptom control. Emerging view. The most modern perspective? Schizophrenia is a circuit-level dysfunction with neurodevelopmental roots, where dopamine and glutamate systems are part of one interconnected network, not isolated players. And that wraps up our discussion on schizophrenia and its underlying mechanisms. I hope this session gave you a clear and connected picture of how dopamine, glutamate, and brain development all fit into the puzzle. If you found this video informative and easy to follow, make sure to subscribe to the Pharmacology Vault so you don't miss our upcoming deep dives into fascinating pharmacology topics. Also, don't forget to like this video and share it with your friends or classmates who might benefit from it. Every bit of support helps us bring you more quality content. Thank you for watching. This is the Pharmacology Vault, and I'll see you in the next video.